Good evening. Thank you, Jessica, for that kind introduction. I'm pleased to be part of tonight's World Affairs Forum conversation. While it's true to say that it is an extraordinary honor to welcome tonight's speaker, Dr. Philip Dormitzer, the word honor isn't strong enough to capture the feeling I have as a frontline infectious diseases physician at Stanford Health. For the past almost 2 years, I've been a hands-on physician caring for COVID-19 patients at Stanford Health and serving on the clinical leadership team overseeing our testing and vaccine programs. I've had the privilege of seeing firsthand people cry with relief when they received their COVID-19 vaccine. I've also experienced patients' sickness, recovery, and sadly death. As a result of the expertise and dedication of leading vaccine research scientists like Dr. Dormitzer, the progress we have made against COVID-19 is unquantifiable. People are living. They're not becoming ill, or at least not as ill. They're not spreading the virus, or not as much. And that is what it's all about, making sure people can live out their lives with their loved ones. Throughout the pandemic, Stanford Health has treated over 1,600 COVID-19 patients and tested and vaccinated hundreds of thousands of our residents. And to be more specific, as of this week, we've administered over 223 tests and over 86,000 unique individuals have been vaccinated through our vaccine site. In the beginning of 2020, we scrambled for PPE, rapid testing equipment, and ventilators when resources were scarce. We tried therapeutic treatments, some of which were effective, others not. We saw the fear and hope in patients and their family members throughout it all. Vaccines miraculously arrived fewer than 12 months into the crisis. It's hard to believe that last December, we were just starting to administer vaccines, including the Pfizer vaccine that Dr. Dormitzer played a leading role in developing to healthcare workers and the elderly. Now, in less than a year, we're on to boosters and vaccinating children, and there are more therapeutics on the short-term horizon. It's truly astonishing. Our scientific community has saved countless lives. Many of these scientists will be noted in history. Dr. Philip Dormitzer will certainly be among them. His expertise has been honed over a very illustrious career. Last Friday, he was named the Global Head of Vaccine Research and Development at GlaxoSmithKline. Prior to last Friday, Dr. Dormitzer served as Pfizer's Vice President and Chief Scientific Officer for viral vaccines, where he played a pivotal role in the development of Pfizer and BeyondTech's messenger RNA COVID-19 vaccine. He also oversaw Pfizer's RNA work on influenza and led the development of Pfizer's respiratory syncytial virus, or RSV, infection vaccines, which are now in late-stage testing. He previously served as the head of U.S. vaccine research at Novartis, where he led projects that included the research component of the Novartis's response to the H1N1 influenza pandemic. Dr. Dormitzer's educational background is equally as impressive. He studied anthropology at Harvard College and conducted field study of the Efe pygmies in the Ituri forest of Zaire. He then completed his MD PH.D. in cancer biology at Stanford University and went on to train in internal medicine at Massachusetts General Hospital. He also had a fellowship in the Harvard Combined Infectious Diseases Training Program. As an assistant professor of pediatrics at Harvard Medical School, Dr. Dormitzer led a structural virology laboratory that determined the structures of the rotavirus neutralization antigens. Dr. Dormitzer's extraordinary commitment to and contributions to global health has benefited us all beyond measure. My gratitude to him, and I'm certain that of our audience, is immense. Will you please join me in welcoming Dr. Dormitzer? Dr. Dormitzer, the floor is yours.
So thank you very much, Dr. Shah, for the very kind introduction, and of course, especially for your service and taking care really on the front lines of the COVID pandemic. I mean, I am very impressed by the physicians, nurses, and other healthcare professionals who put themselves at risk in taking care of patients, and did so from early on when it was even riskier than before the vaccines. I am kind of between things right now. I am now at GSK, but it's my third day on the job. So normally when I give a talk, I pretty clearly represent a group. Recently, it's been Pfizer and now GSK, but I don't really represent anyone right now because I don't represent Pfizer anymore since I no longer work for them. And with only 3 days on the job with GSK, I really can't be said to represent GSK either. So this will not be the usual representation of a company position, explicitly not. And so this will be more of a personal story. Don't worry, it won't be too personal. This is not a support group. It is the World Affairs Forum, and I'll maybe give a bit of a narrative, particularly talking about how my own experience has impinged on areas of world affairs and of sort of broader interest. I started off as an academic working on rotavirus structural proteins, and what I'll recount is maybe touching on 3 key themes. One is the role of personal relationships in what seem like rather esoteric events like dealing with infectious diseases, and in fact, what is the grease that makes the responses go quickly is personal interactions that one has developed over time. I also will talk about some of the perhaps unintended consequences of policy decisions when it comes to infectious disease outbreaks. And I'm sure there was a third key theme here as well, but I'll come back to that later. So while working on the structural biology of this other pathogen, rotavirus, which causes gastroenteritis in young children or diarrhea, it became clear that understanding the structures of some of the proteins that viruses have, the ones that actually work as the devices that get the viruses into the cells, was key to sort of the modern era of vaccine research, where we actually don't just take the molecules nature gave us, but we manipulate them to make them better vaccine antigens. And maybe that's where it starts the theme of the sort of personal interactions, because I moved from an academic laboratory studying the rather esoteric field of structural virology to the vaccine industry through a personal relationship. And that is, it started with an Italian count, Count Armanisi. And Count Armanisi's wife was treated at the Brigham and Women's Hospital in Massachusetts, and he was pleased with the care she received. And he donated a building. And so we have the Armanisi building, but he also started a foundation, the Harvard Armanisi Foundation. And its mission was to bring together Harvard University scientists and Italian scientists. And I was lucky to have a conference from the Armanisi Foundation in the Virgin Islands, of all places. And while I was there, I met Rino Rapoli. Rino Rapoli was the head of vaccine research at Novartis, which now Novartis was not a new company, but the vaccine division was relatively new. And I gave this talk about how I thought engineering viral antigens could lead to better vaccines. And Rino said at the time, well, how would you like to start an institute? Instead of the career prospects of an assistant professor at Harvard are, as it turns out, a little rough. And that sounded great. And so I got on a plane. First time I'd ever flown business class was flown out to the Novartis. It's not just a compound, it's practically a little city into itself. In fact, they moved the border between France and Switzerland to realize the former Chelman's architectural vision for a building that was going to cross from Switzerland over into France. So quite a grand campus. And went up into the big tower where Jörg Reinhardt, who is now I think chief operating officer at Novartis, was. And he had just a few quick questions. So you're supposed to start an institute. How many people have you ever managed? I said, well, I have a technician and a graduate student. And he said, well, you're not running this institute. And so I went back and started work with someone else with much more managerial experience than me. And ultimately I did ultimately run the institute in the end, but that was after about 8 years. And so that was sort of the start in industry. And I sort of never looked back because the opportunities really opened up from there. I learned that people return your phone calls a lot more when you work in a position like that than when you're an assistant professor. And what I really learned was the difference between what I had imagined a pharmaceutical company was and my imagination as a young scientist. Well, it's a whole bunch of scientists with a few people who do other things. And what I learned is that's not it at all. It's a few people, a few scientists. It's not actually a few, it's many scientists, but it's even more people who do other things. In fact, when I went to my first meeting of a scientific management board and realized there were only 2 or 3 scientists in the room. Everyone else was a finance person, a lawyer, a regulatory expert, et cetera. And you realize that what it really takes to take a scientific idea and realize it is a tremendous number of other skill sets. But early on, something interrupted the progression, and that was the 2009 H1N1 pandemic. And that event set in motion a series of consequences that in many ways indirectly led to our ability to respond to now the current COVID-19 pandemic. At the time of the 2009 pandemic, vaccines were almost entirely egg-based. And the way that you responded to a new outbreak was to basically wait by your mailbox. And what you were waiting for was the World Health Organization to, if it was a flu virus, to cultivate that virus from someone's nose. It goes into this elaborate system. There are over 150 laboratories of the WHO worldwide do all of this surveillance. And when they pick something up, it gets passed to a higher level laboratory where it is cultivated in eggs and literally mated to make a vaccine strain, a process that takes months. And you wait for that to happen. But there was kind of a new kid on the block around the 2009 pandemic. Conventionally, the CDC, the FDA, the NIH were the agencies of the government that really would promote this kind of work. But the new kid on the block was BARDA, the Biomedical Advanced Research and Development Authority, sort of the civilian biodefense group. And they had a different sort of vision of a very pragmatic vision where they really partnered with industry to bring things forward. And at the time, we had a contract from BARDA. And that contract was for pandemic preparedness. It involved making a factory for pandemic response. We had, at that time it was Novartis, we had a contract and a facility in Holly Springs, North Carolina to make vaccine. And the head of BARDA at the time, Robin Robinson, called my boss, Reno Rapoli, and said, there's been an outbreak and currently we've seen the first cases in California. You have a contract to respond to pandemics. We think this could be one, respond. And so at the time, the standard was to simply wait for someone to send you a virus, but we had this new idea that we would make the virus ourselves. Now, this was kind of hubristic. At the time in Italy, we had a graduate student who knew how to, what we call rescue flu viruses, that is, start with a genetic sequence, take that genetic sequence and rescue a virus from it. So no one needs to send you the virus, you make it yourself in the laboratory. And we started to do that. And we succeeded. We weren't able to actually synthesize the virus. We actually had to get some RNA from the Centers for Disease Control and start there, not quite at the synthetic step, but almost. We attempted to start at the synthetic step. And then just because you make something like that doesn't mean you can put it in a human being. A lot of things have to happen, but one of them is you have to make it under a very defined set of circumstances called good manufacturing practice, which basically defines the very tight controls that you need to take something that you're not just going to use for an experiment in mice, but actually bring into a human being. We partnered then with the University of Marburg because they had a BSL-4 laboratory BSL-4 laboratories are beyond BSL-3. These are the laboratories where the nastiest of the nastiest bugs are handled. Ebola, smallpox, that kind of thing. And we didn't need that kind of containment, but we needed a very tightly controlled environment to mimic a GMP kind of manufacturing environment. And they had it there. And we had a research facility in Marburg, Germany. The first Nobel Prize went to Emil von Behring. He worked at Marburg building the Behring work. And right across here, that was on one hillside. On the hillside across was the University of Marburg and this very high containment laboratory. And we made that virus, a vaccine virus. And it turned out that on basically the day we succeeded in making it, we received our virus in the mail from the WHO within a day or so. Now, naturally, we went forward with the virus from the WHO. That was the established pathway. We went on to make 3 licensed pandemic vaccines and distribute them. And the response was, in fact, much faster. And this is rarely recognized, but that response is like much faster. We spent less than half the time making that vaccine that it took to make the COVID-19 vaccine because it was a flu vaccine. And we knew how to make flu vaccines. We had to have some innovation, but wasn't nearly the level of innovation that was needed to make a vaccine against a truly new pathogen, not just a variant on a pathogen for which you already had many vaccines already. But that told us something. If we could actually develop a process to make a vaccine in the same amount of time that it took the whole WHO apparatus to use the existing process, well, shouldn't we be able to do it faster next time? So at that point, in that post-pandemic period, we got together with Craig Venter, who you may know, he had led the private effort to sequence the human genome. His laboratories synthesized life essentially for the first time. And we collaborated with him and said, we want to figure out how we take this new science of synthetic biology and turn it to vaccine purposes. And we went back to BARDA, that sort of new kid on the block federal agency, and got funding for a project to apply synthetic biology to respond more rapidly to pandemics. And we developed a process to actually start with simply an electronic sequence that comes through an email. And we said that within 7 days, we will have a seed virus for making a vaccine if you send us a sequence. And the CDC and BARDA got together and they said, that's a test. They said, we're going to send you the sequence of an unknown virus, a flu virus, and prove to us, this is part of the, if you want to continue your funding, you will return to us a virus within 7 days. And they did that. And they didn't tell us what the virus was going to be. As it turns out, they sent us a sequence for H7N9, a virus at that time was not known to cause human disease. It caused bird disease. And we succeeded. Well, as it turns out, on Easter Sunday, 2013, I got a call from a man I'm going to tell you more about shortly, named Peter Bogner. May have been an email rather than a call, saying, in Shanghai, we already knew that in Shanghai there had been an outbreak of a new respiratory illness that was killing people. We have the sequence. It turns out it was H7N9. In fact, it was a pure coincidence, but it was a variation of the test case that we've been given. And in fact, this was now the real world, not a test, but this was the real thing. And the next day in California, we started the gene synthesis that was done at Craig Venter's labs. And over the course of about, within 3 days, and less than that, it was within 2 days, they'd synthesized the key genes. They put it in an airplane, FedEx, shipped it to us at our laboratories in Cambridge, Massachusetts. And within 7 or 8 days, we had synthesized a vaccine version of the virus. Now, that virus had not been released outside of China. The only place that existed in the world was in China, in Shanghai. CDC didn't have it. Nobody else had it. But we synthesized it and we made it in Cambridge. We provided it to the CDC and we said, you may distribute it to anybody for vaccine development purposes. Well, here's where I'm going to take a little digression to start to talk about some policy issues. And I'm going to start with a story of this man that I mentioned, Peter Bogner. He is not the person you would expect to hear about in a story about infectious disease. He was a Time Warner executive, former Time Warner executive. I don't know if he ran Time Warner, but he had a very significant role at Time Warner. And one of the things that he did at Time Warner was figure out how one can make a business out of music file sharing. How you can come up with a system where creators of musical content are willing to post it and users are willing to pay for it. And Peter's the kind of guy who would go to Davos for economic forum and associated meetings. And while there, he heard a talk about some of the problems that we're having in pandemic response, particularly around bird flu. H7N9 is a form of avian flu, but it's not the form that people worry about most. Avian flu, high pathogenicity avian flu is frightening. Mortality rates of well over 90% in those who get it. Fortunately, it doesn't spread that much. But the world felt it had to be prepared, and we still feel we have to be prepared for the possibility that someday we could have an avian flu outbreak, and mortality from COVID, from SARS coronavirus 2 is a couple of percent. Imagine more than 90%. So this is why it's such a frightening prospect. But there was a problem, and that is that much of the surveillance for this 150 laboratories is done in developing countries. And they see their sequences going to companies like the ones I've worked for, who make vaccines that primarily go for people in developed countries. And there was a question of, why should we be sharing our sequences and not receiving anything in return, when in fact it's going to be used for vaccines that we will not see? Now, there are many good reasons but to have a global response, but there is also, there's a point there. And what Peter saw is that some of the same principles that could make music file sharing work could also facilitate sharing of sequence data, which is just another form of information about pathogenic viruses. And I think of some of his own personal funds and his tremendous amounts of his efforts, and now, of course, there's other funding as well, he set up GISID, the Global Initiative for Sharing All Influenza Data. And it is based on a relatively simple principle, a principle of scientific etiquette. That is, unlike some other systems, you have to sign up to an agreement, either as a poster or user of data. It's a simple agreement. And it essentially says that I'll follow good scientific etiquette. If I use a sequence, I will give credit to the person who posts this sequence. I will offer to collaborate with that person. If I'm posting the sequence, I'm posting the sequence without expectation of compensation, but I do expect to be given credit and an offer to collaborate. It's pretty much that simple. And if you don't do it, you will get a call from Peter to make sure that this works. And as it turns out, it has been fantastically successful. In fact, in this SARS coronavirus 2 COVID-19 pandemic, it is by far the leading source of sharing data, and it's tremendously facilitated this response. But going back to 2013, it was GISID, that organization set up by Peter Bogner, where the China CDC posted the sequence of H7N9. And while we were merrily making this virus, getting distributed, I got a message passed to me from the head of the China CDC saying, you are to stop your rude and illegal behavior. And there were editorials about being a biopirate, because we were the accusation is you were pirating this information and you were using it for profit. Of course, you're using it for public health as well. And this is where relationships are so important. You can imagine something like this blowing up and becoming a big issue. But as it turns out, the deputy head of the China CDC was George Gao. He and I were lab mates. We'd worked together. When I had the structural biology lab at Harvard, it was under in the division of Steve Harrison and Don Wiley. We were both. He was Don Wiley's postdoc. I was Steve Harrison's fellow, and we'd worked together. And Peter, in addition to saying this, he mediated this. He said, you guys need to talk to each other. You guys both signed an agreement about how you were going to deal with something like this. And so we literally got together at a restaurant in Boston. And what was interesting is because there was an element of trust and personal relationship, we could understand what was the issue. It was not that anybody wanted to impede pandemic response. It was not that anyone wanted large sums of money. It was very simple. The Chinese scientists who had sequenced that virus and posted it wanted to be given credit. They didn't want this to come across as if, and it would not be accurate to say this, as if they'd been hoodwinked by a pharmaceutical company. They wanted to see that they were part of a global response, cooperating fully with that response and giving credit for what they had done, which is not hoarding the data, but sharing the data. So this came across as something that was very positive, public-minded, and in everybody's interest, including the interest of China. And that was all it took. And again, it was because it didn't have to go to difficult lengths because there were these personal relationships existing at the time. So the issue was resolved, and we carried on. And maybe this is a time to now transition to what happened in this past year. Maybe I'll say a little bit more. Maybe a couple points of interest. So you mentioned I've talked about working for Pfizer and for now GSK. I don't talk about working for Novartis anymore, and the reason is Novartis vaccines no longer exists. It was broken up into pieces and sold off. GSK acquired a lot of it. So, and in fact, when I'm going back to GSK, I'm going back to many of my former colleagues. In fact, Reno, who recruited me from academics and industry, he and I are working very closely together right now. He's the current head of vaccine research and development at GSK, and we'll be working together through a transition. So, again, a key personal relationships coming back. The Novartis vaccines division grew out of a key discovery by a guy named Michael Houghton at Chiron, which was later acquired by Novartis. He discovered hepatitis C, he and his team. This disease was causing liver disease. No one knew the agent, and through a very fantastic piece of science, he discovered that agent. With that discovery, and also Chiron was among the first to sequence HIV, they started screening the blood supply, leading to the control of hepatitis C and having a huge role in the control, not complete, but certainly reductions in rates of HIV. With that money, they acquired vaccine companies. But the company was relatively new, and we had a mission. We were supposed to make a respiratory syncytial virus vaccine, and we had other missions to make vaccines as well. We did not anticipate that we would be doing pandemic response. And in fact, the company was marvelously productive and profitable for a couple of years, 2009, 2010, while we were responding to that pandemic. But we stopped work on the rest of the pipeline to respond to the pandemic, which meant we weren't meeting our other goals. And once the pandemic was over, we hadn't met our goals, and that was that. And so the company broke up. And the benefit is there was a diaspora of the scientists. And so you wind up knowing scientists everywhere because everybody had to move on at that point. And it was at that point that I went to Pfizer and met Katherine Jansen, who I worked for a number of years, who was a wonderful scientist and a fantastic vaccine developer. And we put together a tremendously effective viral vaccines pipeline. In fact, the reason I was brought in there, I had 3 missions to make a respiratory syncytial virus vaccine using this sort of structural engineering that really led to my going to industry, and that is now in phase 3 trials. To restore a viral pipeline, Pfizer acquired Wyeth, which had a wide viral pipeline, but they also had in development a pneumococcal vaccine, Prevnar. And to bring that vaccine to licensure was going to require an 80,000 person clinical trial, a tremendous effort. And the company had to completely focus on that mission, and they had to let their viral pipeline drop. And it turned out to be a very smart decision. Prevnar became one of the most important vaccines for public health out there, and before the COVID-19 vaccine, the most economically successful vaccine as well. But then with that, they wanted to restore a viral pipeline. So my job was to bring the viral pipeline back and to bring RNA technology in. Because the other thing I didn't mention, while we were at Novartis, we also developed early RNA vaccines. A slightly different variation on the technology than the current generation of nucleoside modified RNA vaccines, but we developed RNA vaccines we call self-amplifying RNA vaccines that behave a little bit like viruses but are non-infectious. And in fact, in 2013, when we synthesized the H7N9 vaccine virus to make a conventional vaccine, we took that same synthetic DNA and we made a model RNA vaccine. Now, at that time, we didn't have the what called GMP. It wasn't the vaccine that would be suitable to go on people. It wasn't made under those standards, but it actually had a role in the outbreak response. And I should mention, H7N9 never became a pandemic. It was a pre-pandemic outbreak. There was huge fear there would be a pandemic. So we actually made vaccine. It went to the US pre-pandemic stockpile. So we were prepared if it became a pandemic, but that was one that didn't happen. And that's another key theme, is that for all of these outbreaks that become big pandemics, there are many that don't, because these outbreaks are popping up all the time around the world, and they're difficult judgment calls on how much response is appropriate for what characteristics of an outbreak. We made that RNA vaccine and we used it to immunize mice, which is that we need an antibody against this new variant H7N9, and we used the antibodies from the mice immunized with the RNA vaccine to test that we had the right construct to make the actual vaccine that went into the US pre-pandemic stockpile. So there actually was, in a pre-pandemic outbreak in 2013, the first practical use of RNA vaccines, but at that time as a laboratory tool, not yet ready to actually go into human beings. So now, coming forward to the time at Pfizer, that led to the Pfizer BeyondTech vaccine. Part of the mission was to bring RNA, and our key target was influenza. Influenza remains as a seasonal disease, a disease against which vaccines are an important intervention, but could be much better. There's plenty of room for improvement there. And they remain a key pandemic risk. As bad as the COVID-19 the pandemic is, a high-path avian flu or high-path flu of any animal origin could be much worse. And so we decided that we were going to make a RNA-based flu vaccine, and we wanted a partner to do it. And it was at that point that with a colleague of mine, Julia Lee, we did a survey of the existing RNA vaccine companies out there, and we found BeyondTech. Now, BeyondTech is in Mainz, Germany. They were an oncology-based company that was interested in entering infectious disease, and they had good RNA technology. They could make clinical trial material. They had a wonderful attitude, a data-driven attitude that they were willing to, an attitude that I very much shared, that you can do preclinical testing, but you really don't know how well something works until you put it in people. So you need to go early into clinical trials and explore what's happening. So that's what we were doing. We were preparing to go into clinical trials with an RNA-based influenza vaccine when we got word of this outbreak in Wuhan. Now, Ugur Sahin, who runs BeyondTech, immediately decided that he was going to devote a team to the early stages of generating the initial RNAs and making the early clinical trial material that it would take. And here's where there were real judgment calls. In January of 2020, the outbreak was in Wuhan. It was starting to spread beyond Wuhan. But we had the history of the original SARS outbreak, which was completely contained by conventional public health measures, and the MERS outbreak, the Middle East respiratory syncytial virus, another coronavirus. It's never been 100% contained because it infects camels. So there are still occasional outbreaks in people from zoonotic transmission from camels, but basically it is contained and quite manageable without a vaccine. And in fact, one of my tasks when I started to lead viral vaccine work at Novartis was to shut down a coronavirus vaccine project that had been started at the start of the first coronavirus SARS outbreak and funded by NIH. But by the time we were actually ready to go into clinical trials, everybody, NIH, everybody had lost interest because the disease was gone. And so with that history, the question is, so how do you respond to that? As it turns out, the early stages of a vaccine development are not that resource intensive. You know, a laboratory of people can do it. But to bring out a vaccine that goes all the way to licensure and gets distributed to billions of people, that is a vast effort. And so the discussion we had is, okay, let's start and let's see where things go. And it would be initially BeyondTech was going to continue. And when Pfizer was going to come in with all the capabilities of a global pharmaceutical company, I should probably watch my time a little bit. So I'll try and bring it to wrap it up in a little bit. Was going to take evidence that this was not going to be contained by conventional means. And that evidence came in February when the CDC reported cases in the United States, not many cases initially, but cases of people who had never traveled. They had no contact with anyone who had traveled. They had no contact with anybody who was known to be ill. And this said, this virus is behaving differently. It is spreading asymptomatically, and we can't follow it. Now, it could be contained by conventional public health measures. China, Australia, for a long time did so, but it was clear that the US was not going to accept the kind of very strict quarantine that Australia and China implemented. And in the US context, it was not going to be contained without a vaccine. And so it was at that point that we came in, and maybe I'll tell just maybe one more anecdote and then we'll move to questions. And that was when Albert Bourla, the CEO of Pfizer, made a rather extraordinary commitment. You know, I actually remember the meeting in which he basically said, this thing is out of control, is affecting the community, it's affecting the economy. This is a disaster unfolding. We believe in this RNA technology, and you're going to make, you and your team, you're going to make a vaccine, and you will have any resource that you want. But we're not going to accept failure here. You cannot come back and say, you didn't give me the resources, because whatever you want, you'll have. And in fact, the upfront investment, I would say, and it was a private investment. This was not government funded, although Pfizer sold the vaccine to the government. Research and development was entirely funded by stockholders. It was a $2 billion, publicly disclosed, a $2 billion investment up front. And literally building factories before we had any proof, or not building factories, adapting factories to manufacture the vaccine before we had any proof of concept that it worked, using the corporate jets to fly samples back and forth so that there would be no delays in anything we were doing. Really extraordinary cooperation with regulatory agencies, so we could, instead of the usual phase 1, then phase 2, then phase 3 trial, phase 1, 2, 3 trials, where we just adaptive designs of trials, and really learning all the lessons that we had learned from previous outbreaks and applying them in this circumstance, leading to the very rapid development of that vaccine, and then the massive deployment, so that the projections, and these are the public projections of 3 billion doses in 2021, 4 billion projected for 2022. And of course, as it turns out, although SARS-CoV-2 is a nasty virus and can cause very bad illness, it's a bit of a wimp when it comes to vaccines. It turns out that it turns out much easier to immunize against than, say, influenza. And so it turns out the vaccine worked very, very well. And in part, that's the technology, but in part, and this is something that we didn't know when we started, you didn't know, was this going to be an HIV or a hepatitis C, one of these viruses against which you just struggle to make a vaccine, or one where your first attempt actually works. And fortunately, it turned out that the first attempt worked, and it turns out to be a highly vaccine susceptible virus. And so perhaps that brings us to the current time, and I'll stop going on at this point so I can take some questions.
I have a question about the trial, the physical trial. When you have a volunteer who gets the shot, how do you then expose that person to the virus?
For COVID-19, we rely on natural exposure. So that means you have to immunize many people, because if you're going to observe enough people who receive an infectious dose, you have to immunize a lot of people. But we had very good epidemiologists who were tracking where the virus was. We didn't just want a person; we wanted a representative sample of the community, looking across ethnic, racial, age, even underlying condition groups, so that we would really get a sense of how the vaccine behaved in a population that resembled the people who would actually be receiving the vaccine. Now, there are challenge studies where you actually intentionally infect a person. Not something I wanted to do with this virus, but we have done them for other viruses that are much better understood, like respiratory syncytial virus, where you actually know enough about the virus that you can really be confident that you're doing something safe, and you may cause someone some discomfort, but you're not going to endanger them. I did not feel comfortable, or nor did my colleagues, in taking that approach with this virus. Oh,
Yes, it has nothing to do specifically with the creation of the vaccines, but I'm curious about the policy issue of how private companies sell their product to governments. Rich countries can buy the vaccines, poor countries cannot. Just talk about that general area where we have issues.
And one thing I'll say is I'm primarily a scientist, not a business person, so I'm going to be kind of shallow about this. But the vaccine is being sold around the world, and it is true that it is first rolled out in the countries where it's developed and in the wealthier countries. But at this point, there is tiered pricing. There are large-scale vaccine donations as well. Some of it facilitated by governments, some not. So, of course, there are many reasons why getting the vaccine out more broadly to more population is very important. But it's not a single price for everyone. There really are a series of mechanisms that, I think we all wish they were more efficient in getting the vaccine out more. And I think a key point is that we are producing, our way out of this issue, so that eliminating the scarcity of vaccine to the point where there really will be enough to go around, not just from one company, but from multiple companies. I'm sorry? The supply of this is good, and I'll just use sort of the publicly disclosed projections, and that is 3 billion doses this year, which is going to end soon, and then 4 billion doses next year. And it's an evolving number, of course. So we really are getting to very substantial numbers. And then, in addition, of course, to the Western vaccine manufacturers, there are Chinese vaccine manufacturers and whatnot. So that's just one part of the vaccine supply. So the numbers are approaching the point where I'm hopeful that at some point it will not be numbers of vaccines produced that are the rate limiting steps, but it's actually other aspects of the distribution mechanisms that will become limiting, and that there will be adequate supply so that these other aspects can be worked upon. Yeah.
What about, sorry, what about the variations to the virus that are going on right now, like Omicron, like it's called? Is that something we should be concerned about? And where do you see like life 5 years from now, like everyday life, everyday people? Do you see the virus being a big part of our life as well, or do you see that we are going back to normal? Those are my two questions.
So maybe I'll talk a little bit about the response to variants. And that's an area where this whole synthetic biology has come in a very big way. So now, whenever there's a new variant that we have a sequence for, both a virus is made and a pilot RNA is made for a vaccine on an almost continuous basis. So as these things come up, we're constantly making new variants and advancing them to various stages depending on how serious the outbreak is and how much it looks like it's necessary to change the strain. To date, we're very lucky. No variant that has been tested yet has actually required a change in the strain of the vaccine. Omicron may be different. What we have to say, we just have to get the data. We don't have the data to say one way or the other yet, but it is possible that this will be the one where we do have to make a change in strain. But I think we just have to wait to see the data on that to really know one way or the other. I'm sorry, you had another question. One was how we handle variants, and the other, oh yes,
About 5 years. Where will we be in 5 years?
Yeah, I don't know what the world's going to look like in 5 years. I mean, I do think that this virus is not going away. It's just too widespread, too diverse to actually go away. The history of other pandemic viruses, particularly flu, is that they ultimately become seasonal viruses. They don't go away, but they tend to become less virulent over time as they adapt to humans and as the immune pressure to escape immunity impairs their fitness. So, we will come into an equilibrium living with this virus. It's just a question of what will that equilibrium look like and how much illness will there be between now and then. Seasonal flu continues, but it is nothing like as bad as pandemic flu can be.
Also, we have also speaking of the future, we have a question from the Zoom. Will mRNA technology be useful in addressing cures for other diseases such as cancer? And if so, what kind of timeframes can we look for such advances?
Well, I think RNA has huge promise for other areas as well. I mean, I hesitate to always, I guess I'm a scientist at core. I don't like making predictions. I much prefer to interpret the data in front of me. And so there, in the vaccine, the RNA is used to produce an antigen that produces an immune response. You can use RNA to produce a protein that has an effect, such as when you're missing a protein that you need. RNA can even be used to make changes to other RNA molecules. So there are many ways that this tool can be used. It's extremely versatile. So I think it's very likely that we will find additional uses that actually make their way through to products over time.
What kind of complications you might have come across as you were developing this?
So, first is the ordinary sort of reactogenicity. And the vaccines are on the upper end of reactogenicity of commonly used vaccines. We have the shingles vaccine, which is not more reactogenic than that. But there is some level of reactogenicity. It's more reactogenic than your typical flu vaccine. And that's your typical pain, fever, malaise, which probably many of you probably experienced after having the vaccine. In terms of more rare outcomes, there does seem to be some association with myocarditis. It's very rare and primarily in young men. As you look at this vaccine, it is well within the range of complication rates that you see for other commonly used vaccines. But for all vaccines, you can never say that there's zero risk, but the risk from the immunization is far lower than the risk from the virus. So for the vast, vast majority of people, the risk benefit is way in favor of being immunized. But of course, all these interventions do have some level of reactogenicity.
Booster shot.
Yes.
I think we all probably have them here.
Yes.
Will that be an event in the future where we all annually will get boosters, or do we wait for something to happen and then get a booster?
Yeah, I think the decision to have the third dose booster that really came after observing falling protection. A lot of it came as the Sentinel often was in Israel immunized this population earlier than other places, so we saw the waning first there, but then we saw it in other countries as well, countries that started immunizing later. And I think in terms of the question of whether we'll need booster doses, it's going to be the same thing. We'll have to show that they're needed. Obviously, you don't wait for that to start preparing because it seems likely enough that it's worth preparing, and you don't want to have to start the development. If you wait that long, it's too late. So, it's a sort of continuous preparedness of new strains as they come out. And there is a reasonable chance that more boosters will be needed later, but we just have to watch what happens for people who've had 3 doses before we can say for sure.
Here's a question from the Zoom. Can you tell us, as a scientist, do you have a perspective on the role of the WHO and especially in vaccine equity?
So, the WHO plays a very important role, and it has played a particularly important role in influenza, where it maintains this giant network of surveillance laboratories. And so the WHO clearly has an important role to play. And, it's often an interesting interplay between the role of industry, of national governments, and of the WHO. And it's a complex relationship, obviously. But the WHO certainly has a role to play, as does industry in all of this.
Also, can you speak a little bit about kovacs and its reach?
Yeah. So I know that the companies I've worked with have participated, certainly participating in kovacs, but that's at sort of at the level of distribution and business. That's a little at the well, actually, the kovacs has facilitated some scientific interchange. So I have participated in that, but at the level of distribution, that's not something I participate in. So I don't have tremendous insight there. But the COVAX mechanism has been used as ways to get scientists together so that we share information during vaccine development.
Here's another question. As a scientist, and maybe this is philosophically and maybe this is practically, how do you balance your thoughts and actions related to innovation, like new technology and speed of development, versus risk? How do you think about that conceptually, and how does it play out?
So this is a key aspect of vaccine development, and actually, it's one of the parts that I like most. I mean, the science is fun, but to actually bring something to reality, you have to get not just the science right, but there has to be a development path. You need to be convince someone to fund the development, which is fantastically expensive. You have a huge, compliance network you have to fulfill, a network of rules you have to fulfill, and it's having the scientific insight and the ability to understand sufficiently the areas in which you are not an expert to find the path through, so that the idea actually gets translated. And managing risk and investment is a huge part of that. And sort of figuring out how you can speak to the people who control the investment about the science in a way that you can come to a consensus is I think, really what separates in some ways industrial science from a lot of academic science. And what makes industrial science so interesting is that you have to get the science right, but if you don't get the business and the legal and the development, public health aspects right, you're not going to produce something in the end. And I think you saw during the course of this pandemic just how challenging the non-science aspects of getting a vaccine development project right can be. Oh, yes, and yes, and maybe I won't go into all the details, but yes, some of them are very aggressively targeting vaccine developers. Yeah, a little better since the FBI raid.
One thing I have not heard much about on the news is what kind of immunity do you have if you've been exposed to the disease, the virus, whether you get sick or not, you're just asymptomatic, compared to the vaccine?
Oh, it's a great question. And it does seem that if you're talking about just illness or just vaccine, in this case, at least in the short term, the vaccine immunity appears to be a bit stronger. Now, immunization after illness also can provide a very powerful boost. But this is a case where there are some vaccines that seem to actually outperform actual infections, and this seems to be one of them, which was again something that was quite unexpected. One question I get a lot is, how did you feel when the results of that trial were first revealed? And of course, very good, but it was not just a sense of relief because the vaccine development program had succeeded. It was a sense of relief that the virus itself turns out to be susceptible to immunization, because it could have gone another way. There are viruses like HIV, cytomegalovirus, that people have been trying to make vaccines against for long periods of time, and you can get great laboratory measured immunity after your immunization, but the virus just doesn't care about it. And the virus just evades it and goes on and causes destruction anyway. So it's finding out that that was not going to be the case for SARS coronavirus 2, that if you have a strong immune response, you're going to have a substantial measure of protection. Was a huge relief.
Speaking of the news, if you're a person, not necessarily from following COVID-19 developments, but if you're a person that's interested in learning about the future of vaccine research or the future of developments in mRNA technology, what do you recommend people look for or read? Maybe a step back from the scientific journals, but what could people like us take note of?
That is a great question. I always refer people to the CDC website, because there's a variety of information there. Some of it for medical professionals, but a lot of it for the public. It is well vetted. It is information that can be relied upon. So I mean that is one of the first places I would go for up to date, really solid information. Certainly, there are good journalists who work for the New Yorker and other groups that have done stories on vaccine development, many of which are very good. They all have their own perspective, and you may or may not agree with everything any one of them says. But on the other hand, these are sort of responsible, thoughtful, good writers who are in. Of course, give me a talk here, and the local flavor is helpful. And no one publication is coming to mind, but I think the CDC website is a great place for people to start.
Sorry, there's also a person here. I'm sorry, I'm not going to pronounce the name correctly, but can you relate any of your speaking about innovation in vaccines with innovations related to CRISPR?
Yeah, so I personally have not worked on CRISPR, but in fact, CRISPR technology and gene editing technology can be accomplished with RNA. And this is one of the really promising areas for future work. I mean, can RNA be adopted in a practical way to start to work on genetic diseases as well as infectious disease? That would be very different. And one thing I want to emphasize is the RNA in the vaccine does not interact with DNA. So it doesn't change anyone's DNA, which is one fear that people had, and it clearly does not do that. However, RNA can manipulate other RNA if you design it specifically to do so. So there is work ongoing to try to adapt RNA in a way that doesn't change your core DNA genes, but can alter the messages that come from those genes, so you get expression in ways that could potentially be beneficial. So it's one of the it's not as straightforward as the vaccine approach, so it's not going to come as quickly, but it's one of the promising areas for the future.
Vaccine for young children and infant is going to be available. How long will it take to have a vaccine for young children?
Oh, for young children, yes. So I don't know the exact timeline. But there is work to bring the vaccine down to younger and younger ages. There are trials ongoing, certainly down to 2 years of age at this point. And but because I'm no longer with Pfizer, I haven't seen the latest work. But Pfizer and other companies are now working to try and bring the immunization down below 5 years of age. But since I say I don't see the results right now, I really can't say too much about it.
We have another question from online. You shared a compelling story about how a personal relationship diffused a potential dispute with China that could have interfered with the vaccine's development. Do you think any institutional relationships could be created or enhanced to ensure cooperation between the two countries in the medical field?
Well, that's a great question. And yes. And in fact, there has been, the scientists do cooperate. The relationship with China is obviously complicated right now. And I was just speaking to a colleague of mine, an academic scientist, talking about how for a long time there were tremendous amount of collaboration between China and the U.S. in science. Tremendous numbers of Chinese graduate students and postdocs who train in the U.S. and how there are concerns from even U.S. academics now about those ties, that they may come under suspicion if they have too close ties to China. So I think it is a point of concern that the political difficulties between the countries could have a reflection in reduced scientific cooperation. Historically, there's been a tremendous amount, but it's complicated. Between different, I won't get too deep into it because it's sort of beyond my area of expertise. But historically, the cooperation has been beneficial for both countries. Maybe I'll give one concrete example. There's an RSV. There's several RSV, respiratory syncytial virus vaccines, a virus that causes very severe disease in young children that are in late-stage trials, and it relies on structural engineering of the key vaccine antigen. And the National Institutes of Health, Jason McClellan, Barney Graham, they did that great work. But a key discovery that enabled that work was done at Xiamen University by Professor Zia, where he discovered an antibody that was specific for the right form of the protein for a vaccine. And that work was done in collaboration with the National Institutes of Health. It came to the National Institutes of Health, and it's been translated into vaccines that, if they're successful in their phase 3 trials, could have a huge public health benefit globally. So historically, for vaccine development, and the fact that in these two outbreaks in 2013 and again early in 2020, the China CDC publicly posted those sequences very shortly after they discovered them, so that although there are difficulties, scientific cooperation does continue as well.
To let everyone here know, I think we can have time for about one or two more questions. If we have any from the audience here, and I'll be looking online. We have one question here. Can you say more about the science behind why some viruses are responsive to vaccines and others are not?
Sure. So some viruses, so there are a couple of strategies a virus can have. A lot of viruses are hit and run viruses, like flu, norovirus, which gives you the stomach flu, rotavirus. You get sick, the virus replicates fast, and at some point your immune response kicks in, eliminates the virus, and you're better. And these viruses often rely on both variation and speed. They sort of outrun your immune system. And so giving the immune system a clue as to what's coming through immunization can be very effective. There are other viruses, herpes simplex, cytomegalovirus, HIV. They set up long-term infections, and they learn to live with your immune system. And they often have very fancy ways of evading immunity, even taking advantage of the interactions between cells that occur in the generation of immunity to spread, as HIV does. Or in the case of cytomegalovirus, having many, many proteins that it makes that mimic aspects of your immune system, that disable aspects of your immune system. So there are these two strategies and of course they're gray areas, but to either try and go fast and outrun the immune system, or to learn to the virus learns to counter the immune system and live with the immune system. And that latter class of viruses, the one that, like the saying goes, herpes is forever. Those are viruses that have learned to coexist with the human immune system, and they are very hard to immunize against. There's some exceptions. If you catch them early, the zoster vaccine, like Shingrix. even though people have that virus, varicella zoster virus, latent in them, boosting their immunity with shingles actually manages to contain it. Doesn't eradicate it, it's still there, but it doesn't cause outbreaks of shingles. So it's not hopeless, because there are examples where you can immunize against a virus that's already infected and continues to chronically infect people and prevent disease. But there are plenty of other examples where we just have not been successful.
You've been speaking about global collaboration. When the pandemic started and we had lockdowns and changes in travel, changes in meetings and conferences, as a scientist, what on the one hand did you lose in terms of ability to collaborate that you can't wait to have back, versus what technology, like Zoom meetings or Microsoft Teams or anything like that, what technologies have you used that you're delighted you started using and you will continue after the pandemic?
Yeah, well, we certainly do a tremendous amount, much more remotely than we did before. And I think we can do more remotely than we thought we could. On the other hand, for building relationships, there is, and for really effective communication, I mean, in-person communication really is better. So, I am looking forward to the time when we can meet more again. But that said, we can do so much more remotely than I would have thought.
I just want to be sure. So Pfizer developed the vaccine. It was the first one to successfully develop a vaccine, and they shared it with the world. Is that how it, or was it the other way around? It was developed somewhere in the world, and then it was shared among everyone else, or?
So Pfizer and BeyondTech worked together. So it was a close collaboration and remains a close collaboration. And then there are independent efforts. So obviously Moderna is prominent, and there are many others from China, from other places, from AstraZeneca, of course. So there is, and this is maybe a topic for another day, but there is both cooperation scientifically, and there is competition, and both cooperation and competition have a role in driving vaccine development, as with all medical development. And one can debate a long time what is the proper balance between competition and cooperation. And there's clearly there's role for both with cooperation, particularly in the scientific arena. The sharing of data, of the sequence, I'm blanking momentarily on the name of the U.S. government program. It's not Lightspeed. It was Warp Speed. Although we didn't participate in all aspects of Warp Speed, the scientific exchange through Warp Speed was very useful, and we participated throughout in the scientific exchange aspects of Warp Speed.
So with COVID, a lot of the long-term effects, one of them is a lot of people are experiencing cognitive difficulties. Is that normal for a flu before it becomes a seasonal flu, but when it was maybe a pandemic flu? Is that normal, or is that something different?
I mean, it certainly has been reported with COVID. Although there are rare cases of influenza encephalitis, it is not a typical complaint after influenza. It does seem to occur more with SARS coronavirus 2 than it does with influenza.
I think we have one last question here.
You mentioned synthetic biology. Synthetic biology, is that field in its infancy, or is it further developed along? And
synthetic biology continues to progress. And from it remains a technology that I think has tremendous future potential. I mean, we use it early for viruses because viruses have small genomes that are relatively easy to manipulate. But the Venture Group has made whole mycoplasma from synthetic DNA, which they put it into a cell that then helped it boot up essentially. But I think you look at the creative ferment around this, not only in regenerating existing organisms or microbes, but in making modifications, but also it's just sort of the clever work being done at trying to come up with almost a sort of decision-making circuitry in genes and really design genes the way one would design electronics. We're very early stages, but I think there's tremendous potential there.
Well, I just want to say thank you to Dr. Dormitzer and thank you to all our World Affairs Forum's members and guests. For anybody who would like to stay for a little bit, we have some coffee and water and a little bit of snacks. For those of you who are confident in terms of eating and drinking without masks. Otherwise, you can stay in this room here. But thank you, everybody, for coming. And thank you, Dr. Dormitzer, for your talk.
Thanks for having me.